Hello guys, I am Dr. Isa Khan and you are watching MedicoTube. Our today's lecture on the neurological lesions localization. Very important for the neurology students or the neurology residents. Well, the human brain is a highly complex structure. Each part has the distinct set of the actions, and damage to the each part results in a characteristic and specific chemical manifestations. Specification of these clinical manifestations is very important for the localization of the lesion. I will teach you a very easy way to localize the lesion. So in this brief video we will go through a upper motor neurons and a lower motor neurons lesions. First, let's understand and learn what is a upper motor neuron and what is a lower motor neuron. Then we will go to the lesions. The upper motor neurons are the neurons located in the brain at the brain stem. The body of the motor neuron is located in the primary motor cortex of the brain. The primary motor gyrus the cortex is the command and control center for the involuntary movements. Now let's go with the lower motor neuron. The lower motor neurons are the alpha and the gamma neurons located in the interior horn cells of the spinal cord. Comes out from the central nervous system and connect to the neuromuscular junctions. So now let's understand. We are an example. We will take the example of the bicep muscles, its lower control and upper neuron control. So bicep are supplied by the C6 and C5 cervical region. Let's go to the another slide. Here is the bicep and here is the bicep on the both side. We will take the example of the motor neuron lesions on the right side and the lower motor neuron lesions on the left side. Here is the cross section of the spinal cord. Here is the brain, and the upper motor neurons arises from the cortical area of the brain, cortical and subcortical area. They go through the corticospinal pathway to the interior horn cells of the spinal cord. And another neuron arises from the other side, and same way they go down and they cross each other just below the medulla. Okay, now come here to these muscles. There is a muscle fiber that is called extrapusal fibers. These are the contraction unit of the muscles. They are responsible for the contraction of the muscles, and they are attached to the both side to the tendons on the upper side, and then on the lower side on the both side they are attached to the tendons. And there is one another unit attached to these extrapusal fibers that are called the intrapusal fibers, or the spindle muscle spindles. Okay, these are the sensory unit, and they control the contraction of the extrapusal fibers. They regulate the degree of the stretch in the extramuscle fusal fibers. Okay, and here on the tendons there are the receptors that are called the Golgi tendon organ. We will talk later about this. Now just the anatomical structure here. Okay, these are the Golgi tendon organs. And there is from the interior horn cells the neuron arises to the extrapusal fibers. These are called the alpha motor neurons, and they supply to the extrapusal fibers at the neuromuscular junctions. Now how they work? Very simply, they release the acetylcholine at the neuromuscular junctions and activate the nicotinic receptors, which will lead to the contraction of the muscles. Simply. Now the another fiber arise from the interior horn cells to the muscle spindles. These are the sensory unit in the muscles. They control the stretch, and they send back the information to the spinal cord that what is the degree of the contraction. So if you increase the stretch in the muscle, the action potential will increase in the spindle fibers, and it will send back to the spinal cord, and it will activate the alpha motor neurons, and alpha motor neurons will contract the muscles. So here is another motor neuron that is called a gamma motor neuron. These fibers supplies to the muscle spindles. So the alpha supplies to the extrapusal, the gamma supplies to the intrapusal are the spindle fibers. Let's suppose if we activate the gamma motor neurons, the gamma motor neurons will contract the spindle fibers, and when muscle spindles contract, the more action potential are fired towards the spinal cord, and this lead to the activation of the alpha motor neurons, and this will lead to the activation of the extrapusal fibers, which will result to the contraction of the muscles. So there are many descending pathways. Some are connected with the gamma and some are connected with the alpha. Now here is another fiber that are connected to the Golgi tendon organs. They are stimulated when there is too much stress on the muscles, and they fire through 1B fibers. These are called 1B fibers. They fire through these 1B fibers to the spinal cord and inhibit the alpha motor neurons. These are the inhibitory neurons. The alpha motor neuron is the excitatory neuron, the gamma motor neuron is the excitatory neuron, the 1A is the sensory neuron, and the 1B is the inhibitory neuron. And the reflex of when they inhibit the alpha motor neurons, and this reflex is called as a Golgi tendon reflex. Now same pathway will be on the another side, same the extrapusal fibers and the intrapusal fibers and the Golgi tendon organs and the alpha motor neuron and the 1A fibers and the gamma motor neurons and the 1B neurons. So now, after knowing all this basic physiology, we will go to the pathological lesions. We will produce an injury to the lower motor neuron on the left side, and we will produce injury to the upper motor neuron on the right side. So let's start with the lower motor neuron first. Injury to the lower motor neuron from the soma up to the neuromuscular junctions, anywhere between this pathway, this is called a lower motor neuron lesion. So let's produce any injury. Suppose this is the virus, this is a poliomyelitis virus, let's suppose, for the explanation purpose. So it cut down the nerve here, the pathway is cut down. Now do you think that this neuron is able to contract the muscles, or do you think this neuron is able to produce the acetylcholine and release on the neuromuscular junctions? No, the cell body is cut and this nerve go to the degeneration pathway. We are examining this patient after the injury. Okay, so the neuron will not able to release the acetylcholine. When the acetylcholine is not released, the nicotinic receptor will not activate. When the nicotinic receptor is not activated, the muscles will not go to contraction. This is the simple way. Another way, the acetylcholine is released in two manners. First, it is released by the action potential. When the action potential starts, it releases the acetylcholine. The second, it continuously releases the acetylcholine in a small amount to send the growth signal to the muscles. So it activates the growth of the muscles. It continuously releases the small amount of acetylcholine and they send back the signal to the muscles for the growth. So one thing, we will discuss all these things step by step: mass, power, tone, tendon reflex, and Babinski sign. Okay, so first we will discuss the mass. So here, when the neuron is not able to release the acetylcholine and it's not able to send the growth signal, do you think that this muscle mass will grow? No, it will lose the mass. So in the lower motor neuron lesions, the mass always lost. Okay, 70 to 80% of the mass loss. Now come on the right side and let's produce the injury to the upper motor neuron. Here the injury is produced to the upper motor neuron lesions, and we will see what happened here. Same, we will check the mass, power, tone, tendon reflex, and the Babinski sign. Now as you see, the upper control is lost from the lower motor neuron. Okay, the upper motor neuron controls the lower motor neurons. Okay, so the upper control is lost, but the lower control is okay, it's not lost. So it release the acetylcholine in a small amount for the muscles growth, but the action potential does not occur. So the mass is decreased, but very very less, in 20 to 30% mass is decreased in the upper motor neuron lesions. That's because of the disuse atrophy. When you do not use the muscles, it becomes atrophic. Okay, so let's come to the power. The power is slight loss in the upper motor neuron. Why? As you know that the upper control is the upper neuron controls the lower motor neuron controls, and it's always the inhibitory. In some cases the upper motor neurons are connected to the excitatory, but it's always inhibitory, and it controls the movement of the muscles. So when the upper control is lost, the lower control are not lost. They still work, and they still release the acetylcholine, and there is the power. There is the power in the upper motor neuron lesions, but very least loss in the upper motor neuron lesions, and on come to the opposite side in the lower motor neuron. So when the nerve is cut here and it does not release the acetylcholine, do you think that there will be power? No, the contraction is not occur, acetylcholine is not there, so the contraction is not occur, and the power is gone. Okay, fine. Now come to the tone. The maintenance of tone is controlled by the gamma motor neurons. Here we discussed the basic physiology before. If here with this neuron, the alpha neuron, with the alpha motor neuron, the gamma motor neuron is also damaged. So it is not able to send the information to the spinal cord, and spinal cord is not able to control the tone via gamma motor neuron. One thing. The second thing, if the gamma motor neuron is not injured, it is okay, but the alpha motor neuron is injured, do you think the tone will be there? No. Why? Because when the gamma motor neurons fire to the spindle fibers, okay, it will release it will activate the alpha motor neurons to contract the muscles, but the alpha motor neuron is already damaged, so it will not be able to produce the tone. The tone will be permanently lost in the lower motor neuron conditions. It will be zero, so there will be a placid paralysis. Okay, in the lower motor neuron there will be a hypotonia or the placid paralysis. Now come to the opposite side. On the upper motor neuron, the neuron is damaged here, but the lower pathway is okay, so it overfires because this is controlled by the upper motor neuron. The influence of the lower motor neuron is controlled by the upper motor neuron, so the upper control is lost, and they will be mad. They will go through their own decisions, and they will only decide that what should to do, so they will overfire, and all the pathways okay, so the tone will be increased in the upper motor neuron lesions. Up to now what we learned, that in the lower motor neuron the mass is lost, in the upper motor neuron the mass is not lost, only 20 to 30% is lost because of the disuse atrophy. And the power is lost in the lower motor neuron disease, but the power is very slightly lost in the upper motor neuron lesions compared to the normal muscles. And tone is decreased and lost in the lower motor neuron disease, but it is increased in the upper motor neuron disease. The muscles become rigid. We have the rigid paralysis and hypertonia in the upper motor neuron type of lesions. Now come to the tendon reflex. Do you know how the tendon reflex occur? When we hit the tendon with the hammer, why we have a jerk? It is a simple pathway. Listen, when you hit these tendons, this is not because these Golgi tendons receptors. This is because when you hit this tendon, okay, the tendon becomes stretch, and when they stretch, the muscle spindles also stretch. When it stretch, it sent back the sensory information to the spinal cord, and it said that there is stretch in the muscles. It activate the alpha motor neuron, and the alpha motor neuron release the acetylcholine, and muscles have a transient jerk, a transient contraction in the muscles. So we are on the left side, on the lower motor neuron disease. So when you hit here, okay, you hit here, the tendon stretch and the muscle spindle stretch, and it send back the information to the spinal cord, and spinal cord asks to the alpha motor neuron to release the acetylcholine to have a contraction. But do you think the acetylcholine will release here? Do you think the contraction will occur here? No, because we lost the control. So the tone is decreased in the lower motor neuron lesions, hyporeflexia. We do not have the reflexes in the lower motor neuron lesions. Opposite to that, on the upper motor neuron lesions, when you hit, it send back to the spinal cord through the 1A fibers, and it asks to the motor neurons, and it release the acetylcholine, and we have the tone. So in the upper motor neuron lesions, we have the hypertonia, we have the hyperreflexia, we have hyperreflexes. When you hit with the hammer, you will have the hyperreflexes, brisk reflexes. Now come to the Babinski reflex, or the Babinski sign. First we have to learn the basic physiology of the Babinski, then we will go to the lesions. Do you know why when we scratch on the foot, why we have this sign? It is because when we are walking on the floor, on the earth, it has the scratch on the foot. So when we have the scratch, we make a grip on the floor. So it is the normal. But what happens when there is lesions? Let's take the example first of the lower motor neurons. When you cut the lower motor neurons, the lesions, it will not send back the information to the brain, to the upper motor neurons. So what happened? The Babinski will be down in the lower motor neuron lesions. When you scratch on the foot, the Babinski will be down because the upper control is not present. But what happened in the upper motor neuron? In the upper motor neuron, the Babinski will up. We will have the withdrawal effect, because the brain control is not there, the upper motor neuron control is not there. So we will withdraw our foot. We will not have a grip, we will withdraw. So in the upper motor neuron lesions, we will have the up Babinski sign, or the positive Babinski signs. Thank you for watching. Hope you guys enjoyed, and the next lecture and the next video, we will go for the specific localizations in the spinal cord and in the brain.